about what are the key decision points when sourcing plasmid DNA. This is brought to you in partnership with Akron. For those of you who don't know me, hello, I'm Małgorzata. I'm the head of content here at Phacilitate, and I will be your webinar host today. In this webinar, we will be talking about quality by design in pDNA manufacturing, and we will be sharing lessons learned from a commercial perspective. We have three fantastic panelists lined up for you today, and David Smith from Akron will be moderating this discussion. And just a quick reminder that this is an interactive session, so all questions are very much encouraged. To submit something, please use the question function on your control panel, and we will address these as they come in. And without further ado, I'll hand straight over to our moderator. David, take it away.
Thank you very much. Hello, everyone. Looking forward to a very interactive discussion today. This will not be your standard webinar. We are not going to be using slides. We're not going to be giving you the lecture. Today is a very strong roundtable with a team that has great experience in manufacturing, regulatory, tech transfer, and everything related to cell and gene therapy through the years. I'd like to introduce myself first, and then I'll introduce the team members today. First, for everyone, Dave Smith. I am the President and Chief Commercial Officer at Akron. I've been here about a year. Before this, I worked for 2 years for Helios, which is a Japanese-based company in IPS, which was coming forward with products for the eye, and later for INK. Before that, I spent 15 years with Lonza. I was employee one in bringing out the cell and gene therapy business. And before that, I worked for pharma. And the panel has, again, a very well-rounded background. Sebastian is the first one up. I would like to talk about Sebastian has 15 years of biopharmaceutical experience. He's brought 10 biologics to the market. He's actually built out 2 facilities and tech transferred products from one location into another and bringing up new facilities. So vast experience around the tech transfer and scale up of biopharmaceuticals. Second one member is Ricardo. Ricardo and I have known each other for quite a long time. Ricardo has 15 years of gene therapy experience. He started out with Introgen, which was a gene therapy company pretty far back there, one of the early ones. He was the head of validation and quality. I got to meet him when Lonza acquired Introgen, which was the spin out of their gene therapy company. This was the start of Lonza's CMO viral and gene therapy business. Ricardo worked his way up through that group, first as quality, and eventually as site head for the Houston site, and was the overall help design and build that very large facility there. And the last one is Don Fink. He is now with Dark Horse. Everyone who knows Anthony knows that Anthony chased them and stole them out of the FDA very quickly. Don brings many years, 20 plus years of the FDA, mostly in the cell and gene therapy group. He headed up OTAT. He has been involved in hematologic MSCs. He can give us all the background we want on anything regulatory around the cell and gene therapy world. So with that, you realize that it's a roundtable, very strong team, very diverse, and it gives us an opportunity to really talk about plasmid DNA. What are the requirements, what we should look at for manufacturers, and where should we be looking going into the future? So I think really a starting point is to go to Don and say, hey Don, this is an ancillary material. This is a raw material. What are the general regulations around an ancillary material?
Yeah, thanks, David. So I want to begin for everyone. Remember, now I am ex-FDA. And what does that really mean? That's tons of experience, a lot of knowledge about what's going on. But I am ex-FDA. If anything I say today conflicts with any information or advice you've been given directly from the agency, you just
So I think we have some issues.
Okay, we have Don. We have little technical here.
Yeah.
Stop. You're stuck.
Yeah.
So sorry everyone.
dealing with that.
Webinars are a little challenging. Don, try again.
Okay. Am I live, audible? Thank you.
Yeah.
So basically what I said in nutshell was that as an FDA person, if I say something that conflicts with any direct advice you've been given from the agency, please follow the agency's advice and not anything I might say. And the second piece I want to give is to be cautious about statements made by colleagues or friends or things you've interacted with when they say, oh, we heard from FDA. Because if you haven't heard from FDA, it could be diluted in terms of its accuracy. And I would take those with a grain of salt and make that a C grain of salt, which is very large. What I want to do is also just really quickly put this into a perspective. So we're talking about a plasmid material that meets the definition of a raw material. Now, if that is the case, a raw material is also called an ancillary material, and ancillary materials are not regulated articles by the agency. The agency regulates products. And so that's just a simple translation, and we're going to presume that this material is meeting the definition of a raw material, which is something that is used during the manufacture of the product that you intend to give to subject or recipient. Now, having said that, the agency is very clear that when it makes recommendations, it encourages, it says the word should, and should is a strong recommendation. It's not must or you have to, but should means you want to pursue this. You should use raw materials that are of the highest quality. All right, so that's a decision, a judgment that you make as a manufacturer as to what's going to be a suitable material. We do know that quality of a raw material in the manufacturing process could impact certainly the overall quality of the end product. And we also know that there could be safety concerns depending on poorly controlled manufacturing of materials that are not specifically regulated by the agency. The most important piece I think you want to know is two things. One is I've heard this question, well, can you actually have a GMP product if you use materials that reagents and materials that have not been GMP manufactured as a raw material? And I think it's important to distinguish here, first of all, where is GMP as a grade? The agency recognizes essentially three grades of materials. That would be clinical grade, something's already approved, licensed, vetted, pharmaceutical grade or pharmacopoeia grade, where there's a monograph which says, yes, this material is suited for and recognized for clinical use. And then there's everything else, reagent grade. Notice I didn't say GMP grade. There is no such thing as GMP grade. A reagent or a material, in this case raw material, that's manufactured according to GMP is still a raw material. It's still a reagent grade. It's not anything special outside of those categories. What it does provide, I think, is an assurance of quality and consistency. The most important piece along this to bring this material, regardless of GMP status, into the manufacturing process. It's you have a plan for doing that. You have procedures for doing that. You have tests and assessments for doing that. You look at the vendor, you look at what they're doing in their manufacture of the product. You look at how they're testing and what additional testing you might need to do once that material is brought online, brought in stream, and then vetted for its suitability and use in the manufacturing process. And this would also apply to plasmids, whether they're GMP grade or not. I think that gives sort of a flavor and a background, and I will now let others opine about some of those remarks and comments or move to issues related.
Hey, Ricardo, let's kick off. Sorry, I didn't give you your title now. Ricardo is now VP of Technical Operations for Neurogene. And, Ricardo, as you hear this, hey, it doesn't have to be GMP. Now, as the technical head of operations, and you're worrying about where that material comes from, what are you thinking about when you're thinking about your plasmid for your product?
And really, the first thing it's an evolving paradigm, and recent responses to a recent FDA guidance that came out January this year on neurological disorders, gene therapy for human degenerative diseases, kind of talks into what the agency is looking at, and they have just very small statement that says plasmids used to generate recombinant AAV should be of the highest purity. If the plasmids are manufactured in a multi-product facility, they should be tested for the presence of other contaminating plasmids that may have been co-purified. So the biggest concern from the agency right now as it comes to plasmids is how pure they are, and how likely is there to be cross-contamination from other materials made in the same batch. So when you look at, the CMOs, because most people are going to a CMO to get their product made, you want to make sure that you're looking at what is their product quality with their focus on cross-contamination control. What are the mitigation strategies that they have in place to ensure that the product that was made before is not going to be in your product, and what QC is there to make sure that they identify potential cross-contamination.
So what I'm hearing is purity, purity, purity. I mean, we need to know exactly what's in it. And in a CMO, almost all plasmid CMOs are going to be multi-use. It's how do we prove the separation between those two sites? And I think you and I have worked through a number of proving CMOs have to show separation. Ricardo and Sebastian, you're kind of working to build out this new site in Sarasota. What did you put in place to show that separation?
Yeah, first thing, the most important is the facility should be designed for the purpose. I mean, start with the existing facility and repurpose the clean rooms can bring some headaches in the future. This is one thing. The other thing is to have system in place, a good quality system, a good manufacturing system, a good PD lab, because everything starts in the PD lab. And regarding the different needs that the potential clients can have, I mean, your CMO should cover all these needs. So I think if you have the highest standard, you can cover from the non-regulated or most cell therapy up to drug substance to vaccines. So I think the most important is to choose a CMO that can walk through the different stage with you. I think this is the most important.
Yeah, I think this comes back to things that we looked at in the past. Since a lot of this team came from more of final product, you want to see that separation that you would see in a final product. For me, that means separate air handlers for different rooms, separate very high airlocks, the perfect acceptable cascades.
Exactly.
And then,
containment, unidirectional flow, if it's possible, single-use systems to have a better segregation and less risk of cross-contamination. So all this control strategy will give you some and will de-risk your process and product.
Yeah, from the agency, several good points here. From the agency's perspective, certainly the purity issue is high, and that's a high bar to meet. And sometimes, if it's a non-GMP manufactured material, for example, you as the receiver are going to have to do an ID test. You're going to have to demonstrate. There may be things you don't necessarily have the capacity or capability to do that you're going to rely on the manufacturer for. So that's an important point. And I also totally agree with everything that's said about the facility, the containment, the design, the control, the procedures. But interestingly, if you recent audits, folks, in our our shop have gone out to, you'd be surprised how many people just don't even pay attention to that. By that, I mean, is they cut corners, they skip things, they don't check boxes, and all of a sudden, there's a problem, and the containment is broken, not because the room was designed poorly, the air was handled wrong, but because they carried a contaminant out on them and walked the wrong direction, or went to the wrong place, or carried it in an undetected ways. So those are all great points, and a reliance on the assurance that those procedures are actually vetted and done as they're designed is really important. Can I ask David, I had a question, and I don't know if this is an appropriate place, but I do think we're talking about the difference between GMP and non-GMP. So I want to go to my colleagues here on this, Sebastian and Ricardo. We're getting questions about, hey, can we use GMP like, or GMP light, or GMPS material? So it's manufactured in the same facility, but it's categorized separately. And I'm trying to get a sense for how you would, in a contract manufacturing way, would distinguish between two materials manufactured in the same place, using the same procedures, in the same sorts of largely the same environment, not exactly perhaps, but what is the difference really for the listener between GMP like and a GMP full? How would you describe that?
So
wow.
yeah,
Ricardo, Sebastian.
Yeah, no. Look, right now, the difference, the main difference, one of the differences is the cost difference. The GMP like, or GMPS, or GMP, whatever we want to call it, not full GMP, is going to be a lot cheaper. So a startup company going into a phase 1 product goes into a facility and sees this segregated rooms. They have good processes in place, but they're not bespoke to what you're doing. It's a great option, and an option that most people are using for phase 1, phase 2 clinical trials, where it's not full GMP. I think as the product progresses through the pipeline, you start getting a little bit tighter control. But the statement I read from the FDA guidance earlier didn't say anything about making everything GMP. You have to have the right controls in place. The GMP like has the right controls in place. And you have to assess that for yourself, of course. You have to go into the facility. You walk into the facility and it's like, is it clean? Does it look like it was built in 1970 and it still looks like it was built in 1970? Or have they refurbished and you don't run away screaming? But also, it's the scientific excellence that you have at the facility. Are there scientists within that company that know what they need to look at when they get the starting material? Most of these starting materials come from academic institutions. Academic institutions are like, well, let's just do this and this in this BSc that we just used 2 seconds ago to do another plasmid prep. So making sure that the team at the plasmid manufacturer understands what could be sources of contamination. And also the timeline. A lot of these things, because timelines are critical in everything we're doing, making sure that the team, the plasmid manufacturer is going to be able to meet your timelines is the other critical point when you're looking at selecting your plasmid vendor and associated with your programs.
Yeah. So from our perspective, the GMP light is a commercial denomination. I think it will change depends of the CMO. So I think the most important is to evaluate this with the client and see the risk. Because for me, GMP is not only performing this in a facility, it's having the system in place, qualify your process, qualify your methods. So it's a package of things. It's not only, run some batches in the clean room. That is our impression. So I think it's good to have this. Of course, during the whole life cycle of the process, you will have different qualities. But I think at the end of this is to have a full GMP product, because I think it's more reliable, and in terms of safety and efficacy is going to be the right product.
Hey, Ricardo, this kind of comes back to one of our old days. You know, for me, I always wonder if I give you a plasmid and it gets run through a standard process, you don't really get a batch record, you don't get custom testing, it's just run, right? And then you do that for phase 1. Then when you go to phase 1, 2, you go to phase 3. This is kind of like what we used to think about. People didn't do process development until they got to phase 3. Then they had to try to prove that it had the same product between phase 1 and phase 3. I mean, how difficult is it with plasmids to prove, you know, I have the exact same product? Or should I almost use it as an engineering run? You know, phase 1 almost is an engineering run with a developed process to carry into phase 3.
No, I mean, and that's obviously the challenge. And for programs, companies like ours, where we're treating ultra rare disorders. I know ultra rare is not a statement from the agency, but we still use it. Your phase 1 quickly leads into a pivotal study. So you have to make sure that your phase 1 material, your plasmid, is of the highest quality and purity as possible. So going to, you know, it definitely, you know, it used to be that I remember people early days of, well, for phase 1, you can use research grade. Yeah, that's totally fine. Made in the lab with no air controls or no clean room. Yeah, that's totally fine. That's definitely not the way of the world anymore. That phase 1 study, definitely you want to have your plasmid made in a clean room with controls in place and appropriate, you know, quality systems that support it. Whether you call it full GMP or not, it's as you said earlier, it's a marketing tool.
Yeah, this is from the agency perspective. Absolutely, really want to hone in for the folks listening out there on this expedited timeline development. That is such a critical piece to where CMC is now jeopardizing the success of really what appear to be clinically effective products, investigational products that are coming to licensing stage and can't get across the final goal line because they haven't met all of the CMC requirements for the production of the product. So as an innovator or a sponsor, think about where it is you want to end up, and think about what that timeline is going to look like. Think about these types of questions rather than trying to make these decisions on the fly, have that plan in place. And I agree with you, particularly if you are you intend to make a switch. You intend to start with GMP-like material, highly controlled, good manufacturing process, well tested, well managed, but you just want additional consistency, perhaps, or additional assurance of that, and you want to be able to switch it during development to the GMP version of this product. You will have to undergo an assessment. You'll have to verify that the two give you comparable results. And I can tell you, if you're inside underneath before you get to the licensed product, post-licensure, if you're inside the IND stage and you're using that word comparability, I'm going to speak this on my own. It's a dirty word. It's a dirty word inside. It is almost impossible these days through analytics alone to be able to demonstrate comparability. I mean, that's just the position that we've been seeing in the practice. So you need to think about what that comparability study is going to look like, how much time and energy that might take to switch midstream from one material to another. It's an important question.
Yeah. And let me add something
go ahead.
Sorry, Dave. You have to think that the validation exercise starts in the early stage, in the PD lab. So this is really important. I mean, all the information that we will get in those stages are going to be very useful in the early stage. So I think it's crucial to have this, exercise at the very beginning.
So I'm starting to get questions, which is great. I'm happy to see it. The one just came in. A few years ago, FDA began asking sponsors to provide complete DS modules for viral vectors. More recently, similar requests have been made for the gene editing tools. Can you anticipate the agency will begin thinking about plasmid DNA with similar lens?
Yeah, I think, I think to the extent that it's a piece or a component or a part of a drug substance, I don't, it's a material if it's truly ancillary, a raw material. No, I don't think you're going to need that level of scrutiny specifically for a particular component of the manufacturing process. It gets integrated into the drug substance, and then there's full vetting of the drug substance. I think it's fair to say that, at least in the vector production things can fall underneath that rubric and won't necessarily be parceled out further. I think it just becomes, becomes almost unmanageable within just the process itself. You have all of these things serving as module 3s. You got module 3 and module 3 and module 3 because each one has to have its own verification. I don't know that there's energy for that at this moment. I think it would depend primarily on inability to demonstrate consistency of product, as well as any attendant safety things that arise that apparently are not being capably managed under the current approach. So I'm not sure that it would reflect back on plasmids, and that would be pulled out more as it's been more progressive in terms of understanding what drug substance means and drug product in those compositions.
So, another question, and I'm going to kind of throw this across to Ricardo first, then Sebastian. You're walking into a facility. You brought it up, Ricardo, which is what did they we're getting cell banks or sequences from a research lab from a university, right? You're walking in. What would you expect that CMO to tell you how they're keeping control of all those different banks? What testing would you expect them to be able to tell you they're doing to show that you're not getting a contaminated facility? I don't know if that's the right word, but a facility at risk. How's that?
Right. No. So like I mentioned, the scientific excellence as to what testing is definitely one of the key assessments that you do as you walk into that facility. But also it's knowing, you want to do your Sanger, not just Sanger sequencing, but now it's NGS is so readily available. Just do that full NGS of that cell bank, the initial plasmid stock. Figure out what it looks like, what are the contaminations, what deletions does it have. ITRs are just horrendously bad at just getting deletions all over them. So make sure you test that plasmid like crazy before you go into making that cell bank your master cell bank. Just test it to no end, make sure it's the purest thing, and then walk into the facility and look around. Is it clean? Is it organized? Look behind, look at the mechanical rooms. Mechanical rooms tell you everything you need to know about a facility. If they got a bunch of buckets and mops and all the old equipment that hasn't been, they're just telling you they just shoved it right in there before you walked in the door. So you get a good feeling for how organized a facility is, because that tells you how well they document, and that tells you how well they clean.
Yeah.
My favorite one always was walking into QC, open up the refrigerator, and see how much is out of date. That was always my trigger. Sebastian, what kind of bar are you going to throw up? Or you want to make sure that this facility, the facility you're putting together, stays at top. You want to prove that there's separation. You want to prove that there's no one's bringing in anything that can potentially cause you issues in process development or in the suite. What will you require to bring in stock?
Yeah. Now, first, I want to say that the quality is built, it's not control. That is the main phrase and the main rule of everything. So we are building the quality, not only the quality assurance, not the quality control, also the people from manufacturing, the whole company. So in terms of adding control and have a safer and effective facility, we work in room segregations, different air handed units, again, single-use system. So I think all adding the process, because we are designing the quality during the process. So these factors have good in-process control, different steps. So I think all this package will provide a reliable product to our client. So this is important. I mean, it's not only one part. I think it's the combination of the different things.
Mm-hmm. And I just want to quickly follow up with something Ricardo said, which it also sort of touches on the question regarding Plasmid and any expansion into sort of like a module 3K. You raised a point about the cell bank or the master cell banks that are used to manufacture, produce, and generate the plasmids, and that is captured into 2020 gene therapy guidance from FDA, which when it talks about qualification of your plasmid, it references highly tested, highly characterized, highly qualified cell banks and master cell banks for that purpose. So you go to the core of what really matters, and that's the engine that's going to be producing this material and making sure that it's suitable for purpose, and so that kind of layers in as well. That's a good point about the cell banks.
Yeah, and we are following the ICH guidelines Q5, so for that, so we have the right controls in place for our cell banks. So I think gives another layer of safety and security in our process.
Actually, Sebastian, when I was introducing you, I actually noticed somebody forgot. So you're located today in Sarasota, and what facility are you building out right now? When we keep on referencing the facility.
Yeah, no, we are building, I mean, a very nice facility. I mean, with high level of segregation. I mean, the systems that we are putting in place, complete single-use alternatives for I mean, avoid any cross-contamination. So, I mean, we have, I mean, very nice facility. Also, amazing QC lab, and our PD team. I can tell you that is well experienced. They have experience in microbial fermentation, in purification. So, I mean, these combinations will give some level of security for any client.
So, well, thank you, Sebastian. So, guys, we get to go to my fun favorite question. Okay, tell me about you walked into a facility. What's the thing that you just cringe when you guys have walked into multiple facilities looking at them? What makes you cringe when you walk in and it's just not right?
Well, first, did they answer the door when we knocked? And said, "Yeah, come on in. You're welcome. Sorry you didn't tell us in advance." But actually, these for a license inspection, you would have advance notification. You want to see certainly there's physical attributes that have been touched upon in terms of the structure, and so you want to tick all those boxes, and you want to see demonstration, airflows, compression, air pressure containments in these suites are accurate and are reliable and are the things that are being looked at, and not just statements made. I think that what happens, I think the thing that really, besides the visual and sort of going into a space that is supposed to be a clean room and for high level maintenance, and as we chatted if offline once, talked about, it looks more like a teenager's room where a bomb went off. There's open boxes, there's things not put away, there's papers on top of any surface because you can put them there. That kind of lack of concern for the integrity of that space is easy to see, and it's a clear flag that this probably runs deeper than just this space, and it's probably integrated throughout all levels of personnel and staff that are involved in this process in terms of adherence to rules. So that's probably the biggest piece. I think the other one that is of interest, when you go on an inspection and it's for a product license, you love to look at documentation. And one of the things you love to look at are out of specification. You know, when did something not go right? When did it fail? And you know, there's they have documents and plans that said, yeah, we log all of our OOSs, and you know, we investigate to determine root cause. And so if you go into a facility, say, and this is probably when I was on inspect, one of the first things that a seasoned inspector did, yeah, okay, thank you for your tour of the facility. I want to see your OOS file now. Okay, so you know, you got to go and you got to go get that. And you look at it and you say to yourself, I see here currently over 30 open OOS investigations over a period of 9 months. Hey, why are they open? Why aren't they closed? And what are you doing to address these issues? Because when there's that lack of control of a space where things have already demonstrably gone wrong, something has demonstrably occurred that should be addressed, and it's still sitting on somebody's desk to be done later, that's a concern that ticks a box right away, and you know, it runs right up on the 483 form. Not adequate control of the facility and the process to make sure that it's operating at the level it's supposed to. So those are, you know, those are just some of the things that I hear about and I've seen. To be fair, they'll never let me on the floor. I can't dress in the gowning room, right, without touching 45 objects. So I just stand and watch through a window if I can see that. So I'm not the best person, but those are the kinds of things that a seasoned and experienced investigator notices and sees.
Yeah, but you also mentioned the one you didn't have to get down, which is, can I actually walk down the hallway?
Yeah. Without it being a maze, dodging materials that are just randomly placed because they're easily accessible that way. Well, I just put it there because I need to pull it back right away. Yes, thank you very much for the maze walk. Those need to be organized at least to one side so there's a clear pass through so that you're not potentially disrupting flow and getting in otherwise. Yes, that's correct. Thank you for good remembering. I didn't want to overstate my welcome there, so I do not.
Thanks, Ricardo.
Yeah, no, you know, like I mentioned earlier, it really is the organization, as Don mentioned, you know, the organization, the what is happening around the facility. But you know, to Don's points, it's really digging into those KPIs. How long are deviations open? How long does it take to close a deviation? Yeah, there's some that you're going to have to requalify some equipment. So those could stay, but that can be a kappa as a tied to the deviation. So there's ways of closing the deviations. And a lot of people just leave that deviation lingering on and on and on, and it just tells you everything you need to know. As a former head of quality for the Houston site, I had that always in mind as I turned into the site head of, you know, kappas and KPIs are things that your clients are going to come in and focus on as they're doing their audits of you. So it really is that KPIs are very, very important.
Yeah.
All right. So let's just, well, we're hitting quality system pretty heavy. So what quality systems do you expect to see? You're walking in, you know, it's a batch record on a piece of paper, you know, or what would you say is the standard today for good quality systems? What platforms do you like to see? What platforms do you like to see in the system, in their systems? Oh, sorry guys, Like, is it documentum? Is it, do they
Yeah,
want to see limbs? Do they want to
hey,
see?
Yeah, let me jump in on this, and following the two previous topics, because I think these two topics are related. I think, I mean, a good reflect of your quality system and is how your company manage the CAPAs and deviation. I think is critical, and they are not jumping directly to the root cause, and all the root cause are assigned to training, because this is the most common thing that you can see in CAPAs. So to solve this problem, I think having a good EQMS to make this process easier, I think will be a good feature in your CMO. So I think this is important. Like
There's a lot
TrackWise.
of good options now other than Documentum and other programs. There's a lot of really good quality systems that help track and generate those KPIs that I want to look at. There's ones that do track your training, track your CAPAs, track your deviations, all online, and part of living compliant. So there's a lot of really good tools out there, and you know we're implementing one of them from the start, because the earlier you have it in place, this you can completely capture your entire history of deviations just by going online.
Mm-hmm. You know, one other thing I've been seeing a little bit, and again, this is more for the actual folks that do the business, but some things that have been done is barcoding and having a system that tracks through a barcoding mechanism, all stages and steps, and to the greatest extent possible, eliminating handwritten documents or things that are in notebooks. And you know, even recently, somebody I looked at documents that were submitted that had been scanned. You know, so they had to take it, scan it. It's not, they can't just send me the electronic version. They have to scan it. You can almost not read it sometimes. You know, well, how do I know? I can't even tell what this number is. How do I know if this is accurate? So things that mitigate against inaccuracies related to legibility, you know, and having systems to clearly demonstrate accuracy that are legible and readable and interpretable by whoever's having to do that review is really helpful and strong. And I don't know how that fits into what you've been talking, because I'm not that expertise, but those kinds of things can be challenging, particularly in following longitudinally a process related to any particular product.
Yeah, no, I agree. And also transfer, I mean, data, duplication of data, I think can bring some issues. So have one source of data, I think is important.
And look for those sticky notes all over the place where people report.
Yeah. This incubator's in use, keep away.
The password in the sticky notes. Password for computer.
As long as they're in plastic, that's, you know.
Cool. One question, Ricardo, you were kind of talking about you're in phase one, you're putting a quality system now. Kind of think about that thing. I meant to ask you, Don, if you had to kind of put a mindset. I start thinking in cell and gene these days that there's really almost a pre-pivotal trial and a pivotal trial, and that's about. I mean, what percentage of the industry right now would you say is the two and done almost? Would you say it's 60%, 70%? How much is one pivotal trial going forward for companies?
Yeah, I think there's I found it so interesting, to be fair, just as an observer, the amount of energy and effort that goes into these orphan diseases or rare indications, very few patients, and so one thing I like to say is, to get the data necessary in these sort of indications, clinical trial design, that's relatively straightforward. You do that on a piece of paper, so you can sit there around the table. You can say, we have this rare indication. We're going to have to figure out how we're going to, capture this data, what trial design we're going to do. And you can have an hour, 2 hour discussion, then come back and forth. but wait a minute. What about your CMC colleagues? They're over here going, well, wait a minute. You know, you're going to start collecting efficacy data for a what? So there's this mismatch, really incongruous mismatch between the timeline for a clinical study that could be executed, given there's very few patients or limited numbers of patients, and there may be competition for those patients with multiple innovators. So the trial design features are relatively, I don't want to say they're straightforward, but can be managed in a different, like an order of magnitude different time frame. But the CMC needs to be commensurate in its developmental maturity. Now, you know, if you were to just do canonical, you know, I think if you look at, here's our CMC guidance for phase 1 clinical studies. Well, you know, that doesn't even apply here, does it? Really? I mean, if you're talking about these kind of things, you might do a pilot. Okay, I'll give you that. You might do a 3-6, no more than 10 patient pilot, but even there, you're going to want it. And if the clinical data is strong, we want to capture that. So you're right off the bat, you're not even in phase 1 anymore. You're essentially in a phase 2 or phase 3 immediately. And you're right, David. There isn't the same sort of luxury of flexibility because GMP compliance is required throughout development. But the question is, what is that level of compliance enforced or required? And you know, I heard Sebastian, you talk about single-use items, container system closures, and those kinds of things, which can be brought in to address, compliance issues along GMPs through those kinds of mechanisms. But all of this paperwork, the loggings and the process controls, all of that needs to be mature enough that the data collected in those clinical studies can then be used to support a license, and that's a huge challenge, and it's a higher bar, and I'm not sure. And where does it get to? And I don't know. In the case of, now we're getting off a little bit, but the one thing that hangs up most people right now is the potency or measure of biological activity, and that's true. You need, your vectors, you measure potency in some way with them as well as drug substance and drug product. And if that assay is not identified and qualified, going into a study where the data is meant to support clinical efficacy, you're in trouble. That's just the way it is.
Yeah, and that's the biggest challenge that we're seeing. It really is the, one, the FDA is already telling you, get your process as close to commercial ready for that phase 1, and minimize the changes. Yes, there's going to be changes that you got to do from phase 1 to your pivotal, but there's not going to be a whole lot of time to do that. That said, Don, what you mentioned on potency assay, that's our biggest challenge, is really what is the potency assay strategy when some of these diseases that we're working on, the mechanism of action of the gene is not known.
Right.
And nobody in the world knows what it does. But we know that if we put it into a vector and inject it, these little animals just are rescued. And potentially it's going to happen for kids. But now you got to figure out how to do that in vitro, because I definitely don't want to have an in vivo potency assay.
Right. Correct.
Yeah.
Well, I mean, it's kind of like the phase, it used to be phase appropriate GMP. Phase appropriate GMP went. You're almost starting. You have to do your work early. You have to get it right, because you're going to use the data probably.
Yep. And so, I think the regulatory component to that is as you approach the agency in that pre-submission stage, you get that input up front as going forward. Even if you haven't yet even submitted the IND, if this is your plan and this is your approach, what is the agency's view on the maturity and acceptability of our CMC manufacturing to date, and is it going to be can we use it to support those kinds of studies? You need to know that in advance.
And just guys, I'm not going. I know you guys want the potency assay. I'm not going there.
We're away from that now.
Whole hour. That is an hour or more discussion right now. Because I was on an investor call, and all they wanted to know about was, how do we do potency assays, and does it put all the industry at risk? So that's That's a whole story to go on. Hey, the question came in really for Don and for Ricardo. What is the biggest risk for plasmid production? What's your number one risk?
Cross-contamination. Cross-contamination and the quality of the product. You know, is it going to be, you know, full of dimers or whatever else that's in there? It really is that how clean is it,
right,
and is it going to generate the material that you need?
Yeah. Yeah, I agree with that. The key word there is purity. What is its purity? Is it demonstrable? How did you demonstrate it? If there's any vagaries or variance to what you expect for purity, what impact might that have? Is it impactful? Does it affect either the quality of the output or safety related to the subject? So I agree with that, and that consistency of that preparation over time. You know, assuming that you need to get it, get batches of that on a recurrent basis. How do you ensure that integrity and that consistency of purity? And that's probably a really high bar.
Yeah.
Good one.
Yeah. To do that.
One.
Yeah.
Ahead,
I think
no.
And I think to do that, it's crucial to have a good process characterization, define, which step is removing process and product impurities, work with the binding capacity of the columns and move on those ranges, define the upper and lower limit of your process. That's crucial.
There's an interesting question here, and I'll reword it slightly, but okay, so we're getting early. We're talking about earlier, making sure it's in place, it's right. Okay, so now we're making our GLP product for our path talks. How much control are you looking for? Because you're coming back and filing that data somewhere. You know, what are you expecting from that PD lab is generating your path talks data product?
Oh, do you want a regulatory view first, or do you want to let people that have knowledge go?
Yeah. Let's hear that one for sure.
So the regulatory expectation for developmental studies, particularly in the GLP space for farm talks, is that the process that's being used, and I hate to use I don't want to use the word lockdown. I've heard lockdown, I've heard soft lock. I love soft lock. I don't even know what that means, but the process that you're making the product with those materials needs to reflect what it is you intend to do when you go into the clinical study. It may or may not necessarily be done at clinical scale, but any scaling up should not involve different steps having to be brought in to handle increased capacity or a substance. So it should be, just able to take those implements that are currently used, maybe add more of them, scaling it out, but not necessarily a fundamental change. So you want that material to reflect to the greatest degree possible. That would be, i.e., 100% what it is you intend to go forward in the clinic, and not be some early version or some prototype. And what the farm talks reviewer will do, this is always, you love this. So at the FDA, 3 people are assigned an IND. There's a clinical review medical officer, there's the pre-clin test farm talks, and then there's a CMC person. And the farm talks, they get that section with all of those studies, and there's, this lot, that run, that batch, this one. And the first thing they do is they come down to the CMC reviewer and they say, hey, is that material the same as what they intend to use in the clinic? And I'm like, well, I guess, I'm hopeful. I think that's the objective here. So that material, those processes, the way you test, all of that should be pretty well established to represent what you intend to do to support a clinical trial. Now, is this a pilot animal study? No, I'm not talking about proof of concept data just to support moving forward. I'm talking about critical, pivotal farm talk studies intended to put into an IND submission to demonstrate the safety and proof of concept to support clinical utility.
Yeah, from my side, and quickly, I know we're running up on time, but we record a lot of documents in the PD world, and we are actually moving towards doing that electronically as well to make sure that samples are tracked easier and using electronic lab notebooks, because it is kind of the way the world is moving to be able to have all that data in a more presentable, easily readable format rather than somebody's terrible handwriting in their PD lab notebook.
Yeah, Sebastian, how are you taking care? I mean, I know you guys are thinking about that. What are you guys doing?
No, yeah, you are right, Dave. We have a GLP system in place, but based on the pillars of the GLP: people, equipment, documentation, and of course raw data, because this is the critical part: raw data and source of the raw data. So we are managing this in an appropriate way.
And so since you reworded that question, and so having gone back to Ricardo's really insightful response to the actual question, the documentation and how it's captured and recorded, there's not a requirement. Certainly, you're not compelled to do something in a certain way with electronic capturing. But I think the points all cited, if you can get there, do so. But you want to use a system that's verified and vetted and validated to be accurate. And accuracy is the most important feature of it, and not susceptible to to inaccuracies. You want to make sure that it's reliable.
So, guys, we're out of time. Thank you very much. I appreciate that. It was a great roundtable. I appreciate all your input. Hopefully, we'll get to explore some of those other issues as we go forward. And I want to thank you for your time.
Okay. Thank you.
Thanks, David. And I'd like to extend a huge thank you to the panelists on behalf of Phacilitate. We've covered some really critical points today. And just to close off, this webinar is available on demand. So if there's anything that the listeners need to refer back to, this will be available as a recording in your inbox later today. And to keep up to date with other webinars like this one, you can also always visit Phacilitate Exchange. And with that, I'm happy to close things off and see you at the next one. Good afternoon, everybody.
See you.